So now that we have our messenger RNA strand, we have a full set of directions on how to make just that one type of protein. We've gotten rid of the additional information that we don't need. In other words, the introns have been cut out and the ends of our exons have been pulled together to make a nice, concise set of directions. These directions, which are our messenger RNA, are going to move out of the nucleus and into the cytoplasm so that we can start building our protein. So translation, as it says, occurs in the cytoplasm. We need our messenger RNA strand, which you see in red at the top there, but there's a few other characters that we need to introduce at this point. So we also have what's called the transfer RNA. So this is another RNA molecule, and they've kind of drawn it out to kind of represent it like this. So you're not really seeing the full molecule at all. But this kind of character of it shows you that it has these nitrogenous bases of uracil, adenine, cytosine, and this one, just for an example, has adenine, guanine, and there's a whole variety of different types of transfer RNA molecules. And at one end, they have these base sequences. There are 3 bases at one end, and this is what we call the anticodon end of the transfer RNA. And at the other end, where it looks like it has like a little ball, this is the amino acid. So that's to represent the amino acid. So transfer RNAs help to bring in the amino acids so that we can build our polypeptide chain or the beginnings of our protein. We also need the ribosome. So the ribosome is very important. We've always talked about the ribosome as being responsible for protein synthesis. So this is where it comes into play here. Notice that the ribosome has 2 subunits. It has a large subunit and a small subunit. So I always think of it, it looks like a hamburger bun, where you got the top of your bun and the bottom of your bun. So we need our messenger RNA in translation, a ribosome, and we need plenty of transfer RNA molecules that are carrying different amino acids. So translation, the beginning of it, we say this is the initiation stage where everything kind of comes together. So we have our messenger RNA in red that is going to be sandwiched by our ribosome. So the large subunit sits on top of our messenger RNA molecule, and our small subunit of our ribosome lies underneath our messenger RNA. So we're kind of making a sandwich with our ribosome and messenger RNA. So you can think of the ribosome once again as your hamburger bun, and your messenger RNA is like your hamburger patty. So along the length of our messenger RNA molecule are all these nitrogenous bases that have been exposed, right? So these are part of our nucleotides. And this is how that information is encoded, is in that sequence of nitrogenous bases. So the messenger RNA, besides being spliced, a lot of times we tack on various additional molecules at the end and the beginning of our messenger RNA to protect it as it moves out into the cytoplasm. So we have an area that's called the start codon. So this marks the beginning of where the instructions are on how to make that protein. And then we also have a stop codon at the end. So it's a unique 3 base sequence that marks the beginning and the end of our gene sequence. There is only one start codon, and that is AUG, whereas there's various different stop codons. So we have our ribosome, we have our messenger RNA, and we have marked the beginning of our gene with our start codon. So our very first transfer RNA comes in, and it can bind to our messenger RNA only if it has a complementary anticodon to the codon on the messenger RNA strand, right? So the transfer RNA has an anticodon and the messenger RNA has a codon. So the anticodon is a complement of the codon. So once again, it's our base pairing rules. So if there's an adenine on the messenger RNA, the anticodon would have to be uracil because it's still an RNA molecule. There won't be any thymine at all in an anticodon. So across from adenine would be uracil. If we have uracil, then across from it on the anticodon would be adenine. Across from guanine would be cytosine. So the first transfer RNA molecule has to have the anticodon of UAC. At the opposite end is the amino acid methionine. So this kind of marks always the beginning of our sequence. So how do we know which codons correspond to which amino acids? So you saw that the transfer RNA was bringing in the amino acid methionine. So the codon on the messenger RNA was AUG. So if we look in this chart right here, AUG is right here, and it has the amino acid methionine, and it's recognized as a start codon. Our stop codons are UAA, UAG, or UGA. These are all stop codons, and there's no corresponding amino acid that goes along with them. So this table is how you can look up which codon corresponds with which amino acid. This is not something that you have to memorize at all, but it is very helpful to know how to read this table. So on the left-hand side of the table is the first letter of each codon. Across the top is the second letter of each codon, and across the sides is the third letter of each codon. Okay, so let's see. I don't. So if we wanted to, let's say, find the amino acid for the codon AAA. So A is our first letter, so we would look in this A block here. Let me get a different color. See if we can make this a little bit easier to see, right? Yeah, it's still hard to see, right? But this is our A block. And then we go, our second letter is also A. So we want to go across until we get to that A block and then come down. And then our third letter is A, which is right around there. So we should sit in this block right here. All right. And we see that AAA stands for lysine. Okay. So how about we do one more? So how about we look at CAG? Okay. So CAG. So here's the C block. We want to go to where it corresponds or connects with the A block for our second one. So CAG. So we should be in this block right here. And the G is on the bottom. So CAG is right there. And that corresponds to glutamine or GLN. Okay. And once again, you don't need to know the abbreviations for each of these amino acids or anything like that either. All right, so let's continue on with translation. So what happens next is we've got more transfer RNAs that are going to come in and bind to the next codon. So our next codon is GGG. So the transfer RNA that comes in has to have the anticodon CCC. It brings with it a different amino acid for that codon of GGG. So now we have glycine. The first amino acid moves over, right? So it's moving over and it will attach to glycine. So now our methionine is attached to glycine. So we're starting to build our protein right here, right? So this is our little polypeptide chain, and it's based upon reading off this sequence on the messenger RNA strand. So once the methionine has been transferred over to the glycine, the first transfer RNA molecule falls off. The ribosome kind of slides along, opens up another codon sequence. So now we have UCC as our codon, and the transfer RNA that has the complementary anticodon will come in and bind. So U would be across from A, G would be across from C, and once again G across from C. So this brings in the amino acid serine. So we keep on doing this, keep on moving these over to the next transfer RNA. Once the transfer RNA loses its amino acids, it falls off, and the ribosome keeps on moving and sliding down the messenger RNA strand. So we keep on doing this until we get to our stop codon. So here we have UGA as our stop codon. Remember, there's no corresponding amino acid for our stop codons. So the last transfer RNA would have all the polypeptides or all the amino acids strung together here to create our polypeptide chain. And then there's what's called release factors that come in and bind to our stop codon. This causes the ribosome to break apart, so the large subunit and the small subunit separate. That releases the messenger RNA strand, and the string of amino acids pops off of our last transfer RNA. This string of amino acids can then go on and get folded into its unique shape. Remember, we always talk about structure determining its function. So until that protein takes on its unique structure, it probably won't function properly. So we need to get it folded into the correct shape. Now, a single messenger RNA molecule can be read by several ribosomes at one time. So this messenger RNA can go on to make more of the same type of protein, the same protein, or it can be broken down and recycled.